Hello everyone, my name is Peyton Dodd, and today I'll be teaching you how to use Starterator. The first thing that we want to do is go over some key terms that I will be using in today's video. The first term is a family. Well, a family is a group of similar phages that shares the similar sequences or characteristics. Longest ORF stands for the longest open reading frame, and this is the part of the reading frame that has the ability to be translated. We generally always want to select the start position that gives us the longest ORF. The start codon signals for the start of a gene or can code for the amino acid methionine. The stop codon only codes for a stop of a gene and does not have any other functions. Manual annotations are very important to understand for today's video. Gene annotation can either be manual, which means that they have been viewed by humans, or electronic, which means they have only been viewed by computers. The more manual annotations a gene start has, the better evidence we have for selecting that particular start. Now we are going to dive deep into what Starterator is. Starterator is an online program that examines all the genes within a particular family and generates the longest possible ORF for each gene within a family. Starterator also can give you a list of all the possible starts for a specific gene and how many manual annotations are listed for each type of start. Starterator gives us a graphical representation and a text report of the data generated, and we generally only look at the text report because it is easier for us to understand. The first thing we are going to do is to generate our Starterator report. What you do is you click this hyperlink right here by FAM Starterator, so you click it, and then a new window will open up that generated your Starterator report. So this is the Starterator report for this particular gene in family 55261. And here are all the lists of all the family members, and this first couple of pages are the graphical representation of every start within this particular family. Now I'm going to zoom in so we can see this a little bit better. And so each line represents a different start within that gene, and it's a different start codon each time. Now, not all of these are going to be start codons because some of these will code for the amino acid methionine. So generally the starts that we choose are like kind of up here at the beginning, but that just depends on the particular phage. So the phage that we are using today is SOB. So we are going to scroll down and find SOB, which is right here. And as you can see, there are numerous colored ticks that represent a different start codon. So now that we have looked at the graphical representation of the data, we are now going to scroll and find the text report. Starting with this gene information right here, from here on out is the text report for each start for each gene within this particular family. They are in alphabetical order, so now we are going to scroll and try to find SOB. I have now located SOB's text report. This right here tells you the name of the gene and which gene we are looking. So we are looking at gene 11 in the phage SOB. Its start codon is at location 7,614, and this is just the location of the base pairs for the start. And it stops at the base pair 8,423. So this list of numbers gives you the different candidate starts for SOB's gene. So the start located at the base pair 7,557 only has 2 manual annotations. The start located at base pair 7,614, however, has 235 manual annotations. And the starts at all the other different locations have no manual annotations. So we can use this as evidence to choose the start location at 7,614 because it has the most manual annotations. Now we are going to go back to Pecan and look at the other information. We are now going to scroll down to look at the different gene candidates. We chose to start at 7,614 because it had the most manual annotations and was called by Glimmer and GeneMark, so that is very strong evidence for us to choose this start. The start that we chose does not have the longest open reading frame. However, this is okay considering the very strong evidence of Glimmer, GeneMark, and Starterator to choose the start at 7,614. So the last thing we are going to go over in today's video is how to decide on the correct documentation for Starterator notations on Pecan. This is located in the bar right here, and so we choose a suggested start whenever the information indicates that Starterator aids in the choice of a particular start. We choose not applicable whenever Starterator is not needed or it is an ORFAM. We choose not informative to indicate that Starterator was run for the gene but the output did not assist in the start decision. You now have been given all the necessary information on how to operate Starterator and how to use the Starterator information to choose your start.